Welcome, everybody. My name is Nazar Vasily, and I would like to introduce topic dedicated to the webinar organized by Walter Dandy Neurosurgical Society. I would like also to thank moderator who kindly agreed to moderate this session, Professor James Rutka from University of Toronto, Sick Children Hospital. Um, and This topic is particularly very important. So we will be moving from molecular oncology to translational clinical outcomes. So I would like to start. First, this is just a small recap. I'm sure that everybody is aware glioblastoma is considered the fourth type of glioma, and this is the most common and malignant, with the average 5-year survival rate is just about considered from 7.2 on the average of length of survival up to 15 months. Glioblastoma type 1 and also polycytic astrocytoma and mutational of is considered, and mutation in neurofibrin gene is the most common features. This grade 2 and 3 gliomas, they occur due to mutation of TP53 and alpha thalassemia, as well as X-linked ATRX gene. Um, According to a World Health Organization, glioblastoma is considered as fourth stage glioma and is all seen in the patients over 15 years. But this is the growing evidence that glioblastoma can also develop in children, adolescents, and adults, that these tumors are genetically unique from those that found in older adults. Standard of care is surgery, chemotherapy, and radiotherapy. But despite all of these advances, recurrence is really high. Unfortunately, unmethylated status patients, their survival rate is up to 4 months, despite of Timodal and radiotherapy. Although fluorescence guided surgery was introduced with combination of different photosynthesizers that help to extend neurosurgical resections in patients and improve overall survival after 4 months. Um, this didn't lead to the survival rate that is really expected. So, particularly this study will be, and at this webinar, will be concentrated on the outcomes where PDT is combined and where with photosynthesizer, where we have the light and photosynthesizer, and this combination can cause cell death. However, this was not really clear up the way. So this is not a new topic, and already there are a lot of evidence that shows that up to 40 years where 5LA was studied. Um, However, glioblastoma and cancer stem cells, they highly resistant. They represent a small population of cells within the tumor that can self-renew and differentiate into various cell types. They lead to tumor growth and recurrence. This resistance to conventional treatments, which includes chemotherapeutic agents, including timodal, temozolomide, and radiotherapy, bevacizumab, or etc., there are a couple of factors, including the expression of all the drug efflux transporters, activation of DNA repair mechanisms, and regulation of cell death pathways. So one of the biggest challenges in the delivery of drug to glioblastoma cancer stem cells in their location within the brain, which is protected by blood-brain barrier. So this represents just a significant challenge for the effective delivery of chemotherapy drugs and other therapeutic agents to the site of the tumor. To overcome this obstacle um, to conventional therapy, so according to STOP, there was a study that basically just showed that combination of radiotherapy and plus temozolomide just increased survival rate from 10.4 to up to 26.5%. Additionally, two unique biological characteristics of cancer stem cells, such as their high resistance to apoptosis, their ability to form cancer spheres, and their expression of drug efflux transporters, made it difficultly to effectively target these cells with conventional therapy drugs. And to overcome this, exploration of novel drugs delivery strategies, including nanotechnology based approaches, are just designed to specifically target these cancer stem cells just to improve the delivery of drugs to these cells which are needed. And development of such strategies just they hold the promise. um, but in terms of the challenges and in treating of glioblastoma in its intratumoral heterogeneity, which refers to the fact that these different regions with a single glioblastoma tumor can have different molecular and genetic profiles. Um, this heterogeneity makes it difficult to develop effective treatments that target all regions of tumor. So one reason of intertumoral heterogeneity in glioblastoma is presence of cancer stem cells. These stem cells are thought to be resistant to many forms of treatments such as chemotherapy and radiation therapy, and may contribute to tumor recurrence as well as progression. Research has shown that glioblastoma stem cells are heterogeneous in their molecular and genetic makeup, and particularly contributes to overall intertumoral heterogeneity in GBM. And it also results from the unique microenvironment within the brain, and all these challenges they have been recently advances and the understanding of intertumoral heterogeneity in GBM and the development of the new treatments. It is just aimed to overcome this heterogeneity. They used like personalized medicine, which considers the patient specific molecular and genetic profile. They have big promise. Additionally, the use of combinational therapy, which involves using multiple treatments in conjunction with each other. So, the more effective than using a single treatment alone. However, much work remains to be conducted to fully understand the tumoral heterogeneity of GBM and to develop effective treatments that can target all the regions of the tumor. Um, the one point that which is important is extracellular vesicles, so which can transfer genetic mutations from cancer cells to normal cells, which leads to development of the new subpopulations of cells with the tumor with different genetic profiles. So these extracellular vesicles they can transfer RNA and DNA to recipient cells, which results in changes in gene expression and regulation that can contribute to tumor progression and drug resistance. Well, in another way, it just extracellular vesicles they contribute to GBM heterogeneity by its promoting and communication between cells with the tumor and the surrounding microenvironment. And it can interact with the immune cells and microglia, astrocytes, tumor microenvironment, and modulate behavior, leading to changes in the immune response against tumor. So as well as these extracellular vesicles, they can interact with the extracellular matrix and may modulate physical properties of the tumor microenvironment. They can influence tumor cell behavior and progression. So all these challenges, the role of all of this, the area of investigation and the use of extracellular vesicles and the carriers of therapeutic agents such as RNA and DNA, they offer the potential of development of the new treatments that can target cells within the tumor of the more specific and effective manner. Moving to the standard of care, we expect that temozolomide and radiotherapy, they will induce and make glioblastoma cancer stem cells. They die by apoptosis. However, by using of different um, inhibitors like dasatinib and sorafenib, as well as cabozantinib. Just this is just like regular examples of multi-target TKIs, which has been evaluated in GBM. So although they were improved, TKI, they were improved in GBM mostly due to the lack of success of clinical trials. Was not the way, and they were just restricted across because they were not coming through the blood-brain barrier. So there is a lack of tumor specificity. So another one is just potential for inhibitors like poly-ADP ribose polymerase, PARPs inhibitors, the family of intracellular enzymes that play important role in DNA repair. PARP inhibitors, they block this DNA repair pathway, work specifically well when another repair mechanism is also impaired. So preclinical studies, which is known from PARP inhibitors and GBM, showed encouraging results such as improved radiosensitivity in cells, effectiveness of cetirizine, interstican and the thimazolamide resistant tissues, just reduced the growth of GBM cells and improved survival in animal models. So this which are known like olaparib, talazoparib, and others. Um, so currently they are in investigation in combination with the radiotherapy and chemotherapy, which make antagonistic and antiangiogenic therapies, and which can be combined potentially with immunotherapy. But despite the many efforts, the benefits of PARP inhibitors in GBM clinical trials remain to be determined, which could be attributed to limited blood brain barrier penetration, tumor resistance to PARP inhibitors, and increased hematological toxicity when it's associated with temozolomide. All these integrins are involved in most resistant mechanisms in GBM, and this is becoming the potential of therapeutic target. With several completed and ongoing clinical trials, integrins are the type of transmembrane receptor and the cell's adherence molecules, and they comprise in the two subunits. So particularly they involved in cell intercellular, extracellular matrix adhesion, and they play significant role in just signaling and regulating cell proliferation, migration, and survival. Um, finally, wanted to mention mTOR, which is known as mammalian target of rapamycin, which also proved to be involved in GBM growth and progression. So various mTOR inhibitors are exhibited good in response to preclinical studies, but they until now they have not shown improvements in in studies, either in outcomes as progression of free survival when they use it as monotherapy and in combination with bevacizumab. So, despite these preclinical data, most of this inhibition therapy and combination of the standard of care that can be contributed to poor drug permeability through the blood-brain barrier and to enrollment of the patients with this and without molecular selection. Um, so basically what we are moving we have the challenge because we have resistance and we have highly infiltrative disease, so we are moving to the light activation where a patient can take photosynthesizer just 3 hours before surgery that can be accumulated in malignant stem cells, and which is not only can be used for fluorescent guidance surgery, but it can be used for by targeting residual tumor cells. So basically, PDT plus 5LA was hypothesized to induce apoptosis, but it wasn't clear. So according to different reference and literature sources. So in glioblastoma stem cells, they were just hypothesized that mitochondrial auto-permeabilizations happened and characterized by cytochrome C released, which was basically activated by apoptosome and caspase 9 and 3 cleavage. Um, and apoptotic, this cascade is likely was modulated by BCL2 family proteins and P53, leading to cell death through programmed mechanisms. And at the same time, necroptosis might be activated through receptor interacted protein kinase 1. So the basically this all the issues why it matters, because by overcoming resistant mechanism we can modulate the precision medicine to glioblastoma better in order to improve patient outcomes. So this is all the known advances and the preclinical importance of using navigational setup during 5LA resections. So here we see that the patients took 5LA 3 hours just before surgery, and we can see the tumor fluoresces with the pink, which helps neurosurgeon to resect tumor more easily and precisely. However, this is really difficult to resect all residual tumor cells because they deeper more than 2.5 centimeter, which is invisible by the human eye, and the clinical importance of incorporating all the techniques and additional MRI tractography, intraoperation navigation, the planning of the surgery, they um, critical steps in all of the sophisticated imaging techniques, and basically just helps us just to visualize the brain white matter tracks and avoiding and providing critical information for the neural pathways and which might be at risk, and how to avoid. So basically this PDT and 5LA, they targets the tumor cells specifically, leveraging their altered metabolic profile. 5-MLA, it accumulates preferentially with glioblastoma cells, as mentioned, and was activated at intraoperative elimination. So the ROS formating and induce cytotoxic effect when we apply the light. Moving on, we need the innovation approach that just for recap. We need the light with photosynthesizer. So basically when we have reaction oxygen species upon light, it activates the cell death. So by activating necroptosis pathway, which alternatively to resistant mechanism and apoptosis, it might we overcome and overcome conventional therapies that are expected and resistant to apoptosis. 5-LA in terms is not a new product. This is just the heme degradation pathway, which is currently used in fluorescence guided surgery and widely used in research. It involves just the interaction of photosynthesizer as light, as was mentioned, and typically within the specific wavelengths, which is up from 415 to up to 635 nanometers, which just leads to activation, generation, loss, and subsequent cell death. Um, According to the mechanism of action and application, so PDT leverages like altered metabolic landscape of GBM cells. So when we have the photosynthesizer, when it's combined, when we have the conversion of protoporphyrin 9 due to upregulated heme biosynthesis pathway in tumor cells and upper light exposure with protoporphyrin absorption spectrum. So typically it happens with a 635 nanometers wavelength, which is known upper regulation of specific fluence rate, and the excited photosynthesizer interacts with molecular oxygen to produce singlet oxygen and other ROS. So this initiates oxidative damage. Traditionally, PDT induced cell death, which has been attributed to apoptosis. However, there are a couple of studies that suggested that can be triggered necroptosis in specifically when apoptotic pathways are impaired or can be blocked. So particularly necroptosis here, which is so it can be seen that which is activated when we have the ubiquitination and upon activation of receptor interactive protein kinase 1, upon phosphorylation of RIP1 to phosphorylated of RIP3 and activation of phosphorylated mixed lineage kinase like domain at SRM345. So this is happens when we have necroptosis after we hypothesize when we might have PDT plus 5-LA when caspase 8 is inactive. So basically, in other terms, when we have resistance to apoptosis. So what's happening according to the potential cytotoxic effects? And by looking to the heat maps, which is basically represents the three patient-derived glioblastoma stem cells. So one of them is just methylated, which is G7 and OX5, and I'm OX5 unmethylated and E2 methylated again. So the different timings were just used basically 20, 40, 60, and 80 minutes post elimination, and it just showed this potential cytotoxic effect is seen after exposure to 390 joules fluence rate, which was basically confirmed in 3 biological experiments. And by showing this and to address this inquiry, coming back to G7's glioblastoma stem cells, this is just different assay, but was just used by cytox green, which is just showed that the dying cells within the total count, which was marked with DAPI. So upon binding DNA of DSC cells, cytox green was elicited to green fluorescence signal and facilitated the identification and quantification of cell death events. To investigate all the cytotoxic effects specifically, so very high fluence rate was just used in preclinical model. Conditionally, clinical fluence rate was also used, which was 100 and 160 joules per centimeter square. So it was just to maintain to stimulate therapeutic conditions, and the concentration of 5-LA, which was correspondent and measured to concentration of protoporphyrin 9, which was resected in and measured in resected brain tissue from the patient with GBM, so which was approximately 100 micromolar. Moving on, so we know that, as I said, that glioblastoma, they are the population of cancer cells. They have stem cell-like properties, and they have capacity of cell renewal, and clonogenic assay that just assesses the ability of these single cells to form colonies, and providing the measure of long-term cell survival. So we were trying just to determine whether GCSEs were just can lose their stemness after exposure of light. So we can see that basically, as it shows that um, uh, 160 and 390 joules have cytotoxic effects, and no colonies were formed after 10 days after elimination. However, in terms of the pathway, so in E2 GCs, by using a necrosulfonamide, we see partial dose response and cell death inhibition. By using Inca site experiment with just and by counting cytox green elicited signal. And in terms of apoptosis, which didn't work, and classical ZDEVT FMK apoptosis inhibitor that basically inhibits caspase 3 didn't work, so it's unlikely that apoptosis is involved and is active, unlikely that is activated after photodynamic therapy, and it might be that necroptosis is likely the pathway. So by moving this and doing the protein extraction and in Western blot, we can see after all timings 24 and 60 and 80 minutes after elimination, we see phosphorylated MLKL at serum 345. We do not see caspase 3 cleavage, and we do not see caspase 8 cleaved. But at the same time, we see the same cell line, we see caspase 3 after treatment of BH3 mimetics, which is known BCL-XL plus MCL1 inhibitor, which is a combination of ATP 737 and S63845. Another positive and negative controls were just represented here by Jurkat extract treated with cytochrome C and with no treatment at all. By rigorously evaluate the molecular and selectivity of 5-LA and 5-LA mediated in PDT and in sparing healthy brain tissue, there was the hypothesis that 5-LA and photodynamic and light induces cell death and exclusively in glioblastoma stem cells and leaving healthy brain astrocytes, which represents unaffected healthy brain tissue. So then particularly a fluence rate of 160 joules per centimeter square was used just to hypothesize that it just is clinically relevant, and just to test whether comparative analysis of using mice astrocytes and glioblastoma cells was just conducted. So we see that mice astrocytes were just represented by GFAP and stem cells marker in G7 and OX5, and glioblastoma stem cells were just represented by stem cell marker with SOX2. And so basically we see that there is no cell death after exposure to PDT and 5-LA in mice astrocytes at fluence rate 160 joules per centimeter square, which basically demonstrates that PDT plus 5-LA doesn't affect healthy brain astrocytes. And moving to again to the morphology and this how they die and why it's important, come back, because knowing this we might be overcoming resistant mechanisms. And while even looking for the treatment, so we see, as it was showing, that necroptosis pathway is activated. So necroptosis is very well characterized by cellular swelling, rupture, releasing of contents into extracellular space, while apoptosis just entails cells shrinkage and fragmentation without membrane rupture. Um, Necroptotic cells, they may experience compromised membrane integrity and they display swollen nuclei, where apoptotic cells, they exhibit fragmented nuclei. Necroptosis, they triggers inflammatory response, so via mediated pyroptosis, which is not the case in our, because we won't be because interested in too much inflammation pathway. However, we have release of danger sequence, which is represented by damage associated molecular patterns and which is contrasting with immunological silence if in of apoptosis. So this is particularly important because we have immune system activation after necroptosis, and while we do not have this in apoptosis. So by knowing this, we move to translational neurosurgery benefit for the patient, because we not only overcome resistant mechanism in apoptosis, we have more precision. We can activate with this with PDL1 inhibitors by activation of immune system fight against residual tumor cells in surgical cavity, and by using MRI tractography and neuronavigation intraoperatively just enables to prevent healthy brain tissue damage, and just strategic planning surgery leads to a precisional targeted beneficial outcomes for the patients. This is driven innovation, and by pioneering these new advances in glioblastoma, we have affordability, we have all the centric outcomes that can extend the survival rate. Why we have and moving to other pathways, which is mediated pathways, because necroptosis is characterized by cellular swelling, rupture, releasing contents into the extracellular space. Apoptosis just entails cell shrinkage, fragmentation. Activation of all this necroptotic pathway after PDT5LA and its resistance to apoptotic death events leads to activation of mediated cell death pathway Parthanatos. So to investigate this cell death pathway, there is and to understand this well, we were looking to apoptosis inducing factor which resides in mitochondria, and basically it when it supports the respiratory chain of it, and when it releases to cytosol and translocates to nuclei, this is causes caspase independent DNA damage followed by PDT5LA treatment. And this schematic illustration just portrays the sequential events triggered by might be triggered after PDT5LA. why it's important? Because excessive accumulation of PAR polymers, it can be contributed to mitochondrial dysfunction, and further this exacerbates oxidative stress. And so when once it's released to AIF released to a cytoplasm, and we have AIF in nuclei, we have DNA fragmentation too, so we can have a DNA damage independent of caspases. And again, it's important in unmethylated cell lines, which are resistant to temozolomide, and these patients, they have so these patients, they have survival up to 4 months, and they do not respond to the standard of care. So the fall experiment was just modeled to look for the transition of AF to nuclei, and so basically it's just one of the controls and one of the treatments, particularly 6 minutes after elimination. We are looking for translocation, and so basically cytofluorogram in the study, a published study, just demonstrated that 40% of a correlation was just basically showed up in after for 60 minutes after treatment PDT plus 5LA, and the same time translocation of the signal and AIF, we can see that COX-4 just represented here at Western blot, and the protein were realized that showed that loss of signal in 20, 40, and 60 minutes after PDT, and translocation of AIF to nuclei, which is represented by PCNA nuclei marker. So basically shows that in G7, so we have DNA damage, Parthanatos pathway activated after PDT. So again, this is just the preclinical evidence that shows a small study for in vitro that improve the might be improving the precision and in terms of combinational therapy, and which is important in advancement. The next chapter will be released for in vivo and using stereotactic techniques in mice, with potential of using fiber optics and for deep penetration, for more deeper targeted penetration by using and comparing this with standard of care. So I would like to thank everybody, and again, I'm very thankful to Walter Dandy Neurosurgical Society for organizing this meeting, and for Professor James Rutka, University of Toronto, for joining us at this session. So thanks everybody.